Hey everyone, welcome back to The Explainer. So today we're unpacking a literally groundbreaking paper from a team over at Stanford. It was published in Science in 2026. And look, what we're looking at today isn't just some tiny incremental step. This is a discovery that actually rewrites a foundational rule of biology, stuff we've been teaching in classrooms for decades. So let's just dive right in. Okay, think about this for a second. Can a biological machine build DNA without a blueprint? I mean, ever since we discovered the double helix, the answer to that has always been a hard, absolute no, right? DNA is the ultimate blueprint of life, and to make it, you always need an original document to copy from. Well, until now. Today, we're going to look at a microscopic machine that completely defies that logic. Section 1, the biological arms race. Now, to understand exactly how this rule got broken, we have to travel down into a literal microscopic war zone. See, this discovery wasn't made in a vacuum. It was found in the eternal billion-year-old battleground between bacteria and the viruses that constantly hunt them, phages. As a quick refresher, just think of a standard reverse transcriptase, or RT, as a biological photocopier. It takes an original RNA strand, reads it, and manufactures a matching strand of DNA. The absolute key here is the template. Without that original RNA document feeding through the machine, the biological photocopier just can't print anything meaningful. It needs the instructions. But, bacteria are incredibly clever. Over millions of years of this arms race, they've actually evolved these highly divergent, frankly strange versions of these photocopiers. They're called defense-associated reverse transcriptases, or DRTs. Basically, they use them as secret weapons to fight off viral infections. And that brings us right to the specific system these Stanford researchers decided to look into. Section 2, breaking the DNA rules. All right, let's see how this particular bacterial weapon flips everything we know totally on its head. So the golden rule in biology is super strict. DNA polymerases are template-directed. They require an original document, either RNA or DNA, to dictate the sequence of the new DNA they're building. If you don't have that paper template, the biological copier just jams. It simply cannot work. But meet the culprit of our biological mystery. It's a system called DRT3. It's found in certain strains of E. coli, and this specific defense system takes that golden rule and just throws it entirely out the window. No joke. It synthesizes complex, specific DNA in a way that scientists have literally never seen before. Section 3, meet the DRT3 machine. So what does this rule-breaking molecular machine actually look like? Let's unbox it and see how it operates. Wow. Okay, looking at this high-resolution cryo-EM structure here, you can really see the sheer beauty and complexity of the DRT3 complex. It forms this massive, incredibly stable ring-like structure. Scientists call it a D3 symmetric hexamer. To put it simply, it's this beautifully arranged six-sided molecular engine that relies on some really intense structural coordination to even function at all. Now, if we pull that engine apart, we find it's built from a really strict ratio of three distinct components. First, there are 6 copies of the first reverse transcriptase, called DRT3A. Then there are 6 copies of the second reverse transcriptase, called DRT3B. And finally, you've got 6 copies of a non-coding RNA molecule that just threads right through the whole complex. So these 3 pieces work perfectly together to create a single, highly coordinated immune weapon. Section 4, the protein blueprint twist. Okay, here is where we get to the absolute most mind-bending part of the research. We're going to uncover exactly how the 2 different halves of this machine actually work together to build DNA. So check this out. On the left side, we see the DRT3A half of the machine, and it acts pretty normally. It grabs the RNA molecule, reads it like a standard template, and prints a strand of DNA made of repeating G and T building blocks. It's functioning exactly like our biological photocopier should. But look at the right side. This is DRT3B. When the researchers mapped its active printing site, they realized the space where the RNA template should bind is totally obstructed. Like, there is absolutely no nucleic acid template inside it. 0. Yet somehow, through this completely unprecedented biochemical pathway, it perfectly polymerizes a complementary DNA strand of repeating A and C nucleobases. I mean, how is that even possible? Let me actually read a direct quote from the researchers to emphasize just how wild this is. DRT3B synthesizes a complementary protein-primed poly-AC strand in the complete absence of a nucleic acid template. Guys,
a sequence-specific DNA strand is being built without nucleic acid instructions. So how is the machine knowing what sequence to print if it isn't reading a template? It makes no sense, right?
Well, think of it like this analogy. Instead of being a photocopier reading a flat piece of paper, D3B is more like a 3D printer that uses its own mechanical gears as the mold. The researchers discovered that specific amino acids within the protein itself, like this one called glutamate 26, they actually physically jut right into the active printing site. These protein side chains form incredibly precise geometries. They establish these strict hydrogen bonding networks that perfectly mimic the interactions of an RNA template. So the machine is essentially using its own physical three-dimensional body as the blueprint to force the DNA sequence to alternate strictly between A and C. Honestly, this is the first time we've ever documented sequence-specific, protein-templated DNA synthesis in biology. It's a real game changer. Section 5. Triggering the defense system. So let's pivot back to that biological arms race for a second to see why this DNA 3D printer was built in the first place.
This entire system is basically a giant alarm bell just waiting to be tripped. Here's how it stops a viral infection, step by step. First, a virus called phage T1 attacks the E. coli and injects its own proteins inside, specifically one called ST61. Second, our dormant DRT3 machine detects this viral ST61 protein and immediately springs into action. And finally, both halves of the machine fire up. DRT3A uses the RNA template, and DRT3B uses its own protein body as a template. Together, they start printing massive, complex strands of alternating double-stranded DNA. This sudden flood of weird, unusual DNA completely halts the viral infection dead in its tracks and saves the cell.
Okay, let's pull all this together. Over on the right, you can see the virus attacking and injecting that ST61 effector. In response, our beautifully symmetrical hexamer machine kicks into gear. The top path there maps out the normal RNA templated extension we talked about, but the bottom path shows the newly discovered, totally rule-breaking protein templated extension. Working in perfect concert, these two distinct active sites coordinate to create a highly specific double-stranded DNA product that totally shuts down the invading virus. They are literally building a physical barrier out of DNA, and it's generated essentially from a protein blueprint. Section 6. Rewriting the biology textbooks. Because let's be real, discoveries like this do not happen every day. So what you should really take away from all this is that biology is far more creative than we usually give it credit for. This matters big time because it's the very first time we have ever observed a protein acting as a sequence-specific template to build DNA. It radically expands our understanding of how life can process and store biological information. It proves that information doesn't just flow linearly from DNA to RNA to protein, right? Sometimes the protein can actually dictate the DNA. And finally, it just reveals a completely unconventional, completely novel weapon in the microscopic world of bacterial immune systems. We've really only scratched the surface of the incredible biochemical data they mapped out in this study. If you want to dive deeper into the molecular details and see the experimental methods that actually led to this breakthrough, I highly, highly encourage you to check out the full source paper. It's called Protein Templated Synthesis of Dinucleotide Repeat DNA, published by Deng and colleagues in Science. Honestly, it is an absolutely fascinating read. But before we wrap up, I want to leave you with one final provocative thought. If nature can evolve a machine that essentially acts as a 3D printer for custom DNA, using just its own protein structure as the mold, how could we one day harness this? Could we engineer our own protein templates to print custom DNA on demand? Could this revolutionize medicine and biotechnology? The possibilities are pretty much endless. Thank you so much for joining me on this explainer. Keep asking questions, and I'll catch you next time.